Poliovirus infection. Poliovirus shows tissue tropism by specifically combining with neural cells. It recognizes a receptor present on the anterior horn cells of the spinal cord, dorsal root ganglia, and motor neurons. On combination at these sites, the poliovirus causes destruction of the motor neurons, anterior horn, and brain stem. The destruction of motor neurons in the spinal cord results in flaccid paralysis, Polio Myelitis. Paralysis is not caused by the virus infecting the muscles. There are 3 poliovirus types, with 85% of the cases of paralytic polio caused by type 1. The poliovirus also infects the brain stem, causing bulbar poliomyelitis associated with respiratory paralysis. Do you remember the iceberg concept? Asymptomatic illness is far more common than the clinical manifestation. At least 90% of poliovirus infections are asymptomatic. Abortive poliomyelitis occurs in approximately 5% of infected people. The patient has only the minor illness characterized by fever, malaise, drowsiness, headache, nausea, vomiting, constipation, and sore throat in various combinations. Nonparalytic poliomyelitis or aseptic meningitis occurs in 1% to 2% of patients with poliovirus infections. The patient with a non-paralytic form has stiffness and pain in back and neck. Paralytic polio, the major illness, occurs in 0.1% to 2% of persons with poliovirus infections, and is the most severe outcome. Patients and asymptomatic carriers both can be the source of the infection. Outbreak of poliomyelitis. The polio is a disease with long history. The wall painting shows an ancient Egyptian with polio. The disease is first described by Michael Underwood in 1789. A severe outbreak in the US in 1952. There were 21,000 paralytic cases reported. If there were 21,000 paralytic cases, how many polio infectors that year? Yes, about 1,000 times of the paralytic cases, maybe 21 million. Most of them were asymptomatic. And now, global eradication in near future. Questions are, how can we eradicate an infectious disease in the world? You are right, the vaccine. What makes us have the confidence to eradicate the polio? Key facts about poliomyelitis in WHO website. Let's see the highlighted numbers. Poliomyelitis mainly affects children under 5 years of age. 1 in 200 infections leads to irreversible paralysis. Among those paralyzed, 5% to 10% die when their respiratory muscles become affected. Host response to poliovirus mostly depends on the humoral immunity. Both the serum and the secretory antibodies play an important role in providing protection against poliomyelitis. Serum IgM antibodies appear within a week of infection. IgG antibodies, which develop subsequently, persist lifelong. Secretory IgA antibodies provide mucosal immunity against the virus by preventing intestinal infections and by preventing shedding of virus. Cell-mediated immunity plays little or very insignificant role in the immunity against poliovirus. The host immunity is permanent and type-specific. The virus in the brain and spinal cord is not influenced by high titers of antibodies in the blood. So polio vaccine should be given before infection. Summary of pathogenesis and immunity of poliovirus infection. Source of infection: patients and asymptomatic carriers. Route of transmission: fecal-oral route. Season: summer and fall. Immunity: permanent, type-specific. The virion of enterovirus. Enterovirus is a member of the family Piconaviridae. Pico means 1 trillionth, 10 to the power of minus 12, very small. Then RNA refers to RNA virus. Viridae, the family name. It is a very small RNA virus family. Piconaviridae contains 9 genera, including Enterovirus, Rhinovirus, Hepatovirus, and so on. Rhinoviruses can cause common cold, including more than 100 antigenic types. They are labile at acidate pH, and the optimum growth temperature is 33 Celsius, which are different from enteroviruses. The enteroviruses are very resistant to harsh environmental conditions, sewage system, and the conditions in the gastrointestinal tract, which facilitates their transmission by fecal-oral route. Hepatitis A virus was originally classified as Enterovirus type 72, but now assigned to a separate genus. Diagram again. Piconaviruses are positive single-stranded RNA viruses, very small. Structure of piconaviruses: naked, 28 to 30 nanometer in diameter, RNA core and capsid. Do you remember the homework last week? Can you answer the questions? How many phases? 20 phases. What is pentamer? 5 units. How many pentamers? 12. The capsid of piconaviruses is icosahedral, 12 pentamers. Viral proteins VP1 to VP4. VP1 binds to the receptor. Surface proteins VP1 and VP3 are major antibody binding sites. Internal protein VP4 is associated with viral RNA. The corn, positive sense single-stranded RNA, linear, infectious. The naked picornaviruses genome resembles a mRNA and is sufficient to infect a cell. It has a poly-A sequence at the 3' end and a small protein, VPG, viral protein genome linked, attached to the 5' end. The poly-A sequence enhances the infectivity of the RNA, and the VPG is important in packaging the genome into the capsid and initiating viral RNA synthesis. The genome encodes structural and non-structural proteins. In addition to the capsid proteins and the VPG, the picornaviruses encode at least 2 proteases and an RNA-dependent RNA polymerase. Replication. 1. Receptor. VP1 binds to the receptor, and the VP4 is released and the capsid weakened. The genome is then injected directly across the membrane. 2. Genome is an mRNA, can be used to translate to proteins directly. 3. Virus replicates in cytoplasm. 4. Most enteroviruses are cytolytic, replicating rapidly and causing direct damage to the target cells. CPE. When the viruses are cultured in the living cells, the viruses induce CPE. Left, normal cells. Right, cells infected with poliovirus. Many of the degenerated cells have fallen from the glass surface. Summary of structure and composition of enteroviruses. Viron, icosahedral, 28 to 30 nanometer in diameter. Composition: RNA 30%, protein 70%. Genome: positive sense single-stranded RNA, linear. Infectious: contains genome-linked protein, VPG. Proteins: surface proteins VP1 and 3 are major antibody binding sites. Internal protein VP4 is associated with viral RNA. Envelope: none. Replication: in cytoplasm. Now, have a break. We will focus on the lab test and prevention in next session.